Well, we're going to be talking about chromosomes. This diagram illustrates two different ways that chromosomes are sometimes drawn to represent the ways chromosomes exist in different parts of the cell cycle. Sometimes when chromosomes are drawn, they're drawn like this, with two arms separated by a constriction called the centromere in the middle. Other times chromosomes are drawn like this on the left, where there are four arms connected by a constriction called the centromere in the middle. Why do we draw chromosomes in those two different ways? Because of the way they exist in different parts of the cell cycle. Now, in the cell cycle, you may remember that the M phase stands for mitosis, or cell division, when a cell divides. G1 represents the time period when the cell is getting ready to replicate its DNA. The S phase is when DNA replication actually occurs. And G2 is that time after DNA replication, but before the cell divides. We represent chromosomes in G1 as it is drawn over here on the right-hand side of the slide. We believe that each individual chromosome contains one long linear molecule of double-stranded DNA from one end of the chromosome to the other. Now, that DNA is all packaged with histones to make nucleosomes. It's all packaged with some other components to make chromatin, but there is one double-stranded DNA molecule. That DNA is replicated in the S phase, and so in G2 we now have two double-stranded DNA molecules in our chromosome. The two DNA molecules have not become completely pulled apart. They're still stuck together at the centromere, and the two different DNA molecules have become associated with histones and other components to form chromatin. But we now have just one chromosome, but we say that that chromosome consists of two chromatids. So each half of this would represent a chromatid or a sister chromatid that would contain one of the DNA molecules. So a chromosome, well, and a whole cell will have twice as much DNA in it in G2 compared with what it does in G1. So this is the reason we will sometimes see chromosomes drawn in these two different ways. Now, when we perform a human karyotype, that is, when we look at human chromosomes in the microscope, we do that at the metaphase stage of cell division up here in the cell cycle. We choose metaphase because that is the time in the cell cycle when the chromosomes are the most highly condensed and the easiest to see, the most visible in the microscope. And at that time, at metaphase, chromosomes look like this over here on the left, with a pair of sister chromatids. So when you see a human karyotype, each individual chromosome will have a pair of sister chromatids. Now, in your book, you have two representations of chromosomes. At the top of the page in your book, you've got an idealized drawing of individual chromosomes. And at the bottom of that page, you have an actual photograph of real chromosomes. The artist here has drawn just one copy of each chromosome. And notice that each chromosome is drawn as a pair of sister chromatids as it would exist in metaphase. Now, normal humans have 23 pairs for a total of 46 chromosomes. The first 22 pairs are referred to as the autosomes, and a normal individual will have two copies of each of those autosomes. The 23rd pair is the sex chromosomes, and an individual will either have two X's if the person is female, or XY if the person is male. So 23 pairs, a total of 46 chromosomes. These chromosomes can be distinguished from each other by an expert in cytogenetics by several of their characteristics. One characteristic is size. You'll notice that the autosomes are numbered in order of decreasing size. So of the autosomes, number 1 is the biggest, number 22 is the smallest. A second feature that can be used to distinguish chromosomes from each other is the position of the centromere. The centromere is this constriction that separates the shorter P arm from the longer Q arm of a chromosome. That centromere can be in different places on different chromosomes. So for example, for some chromosomes, such as chromosome number 1, the centromere is approximately in the middle. We would call this a metacentric chromosome. For some of the other chromosomes, such as, for example, 8, 9, and 10, the centromere is clearly offset from the middle. We call those submetacentric chromosomes. And for a few chromosomes, such as numbers 21 and 22, for example, the centromere is very close to the telomere, that is the end of a chromosome. We refer to those as acrocentric chromosomes. So the position of the centromere can help an expert distinguish one chromosome from another. Another major feature that an expert will use is the banding patterns on chromosomes. You'll notice that in this picture, each individual chromosome has its own characteristic pattern of black, gray, and white bands in the picture. Those bands are artificial. They are put on there by staining the chromosomes by one of a variety of different kinds of dyes or stains. Each individual chromosome will take up a characteristic pattern of that stain, and that banding pattern can also be used to distinguish one chromosome from another. So an expert in cytogenetics will break open a cell at metaphase, spread out the chromosomes, stain them, take a photograph, and will arrange the pictures of each individual chromosome in a pattern such as this one to display the human karyotype. In this picture, you'll notice that there are 22 pairs of autosomes, and then in this case, an X and a Y. So this is a karyotype for a normal human male.